I want you to take a moment to reflect on your daily morning routine. Generally, you most likely get out of bed, get yourself dressed, and prepare yourself a quick breakfast to energize you for the day to come. Along with your breakfast, you might have a glass of milk or orange juice or maybe a cup of coffee. However, did you realize that this cup of coffee not only stimulates your senses needed to get through the day, but it also may be causing you to lose weight? In this video, we are going to investigate the lipolytic and anti-obesity effects of caffeine to determine its possible implications as a weight loss supplement. To start, caffeine is a naturally occurring substance that is widely consumed in a variety of forms through a variety of sources. Of the various exogenous sources, approximately 75% of caffeine is consumed in the form of coffee, which makes the stimulant easily accessible to the average person on a daily basis. With such a wide prevalence of caffeine consumption, there is a large portion of the world experiencing its physiological effects, all of which are made possible through the intrinsic properties of the caffeine molecule. First, caffeine is a lipophilic substance, allowing it to dissolve and combine with lipids and fats. This property grants the molecule the ability to cross most membranes within the body, including the blood-brain barrier, which allows it to readily affect the central nervous system. In regards to its metabolism, caffeine is a molecule of the methylxanthine class that is broken down into three metabolites: paraxanthine, theobromine, and theophylline, and this breakdown is catalyzed by the cytochrome P450 enzyme system within the liver. Once metabolized, caffeine has shown to have many physiological effects, including stimulation of skeletal muscle, the central nervous system, and lipid oxidation. In this video, we are going to take a closer look at this fat oxidation, or lipolysis, and identify the possible mechanisms in which caffeine may lead to weight loss. To start, we're going to provide you with a quick background on lipolysis. Lipolysis is the enzymatic process in which triacylglycerol stored in the fat cells of our adipose tissue is hydrolytically cleaved to generate a glycerol molecule and 3 free fatty acids. This process is primarily catalyzed by the enzymes adipose triglyceride lipase, monoglyceride lipase, and hormone-sensitive lipase, all of which are under tight regulation to only function when lipid metabolism is required. One of these regulatory pathways includes the epinephrine cascade, which occurs on the plasma membrane of the fat cells within our adipose tissue. When epinephrine is released from the adrenal glands, it binds to the beta-adrenergic receptors of the adipocyte, triggering a conformational change in the G protein. This then activates adenylate cyclase, which synthesizes cyclic AMP. Subsequently, this cyclic AMP activates cyclic AMP-dependent protein kinase, which phosphorylates hormone-sensitive lipase, converting it to its active form. This in turn leads to an increased rate of lipolysis, and thus increases plasma free fatty acid levels. The free fatty acids released from lipolysis can then proceed to function as essential precursors for membrane and lipid synthesis, or may subsequently undergo beta oxidation in the mitochondria to generate ATP and provide the necessary energy for our bodily processes. In a 2005 study done by Hattori and colleagues, it was shown that the intake of caffeine at 0.025, 0.05, or 0.1% for 21 days progressively reduced the body fat mass and fat percentage in SD rats fed on a high-fat diet. Caffeine also increased the serum concentrations of catecholamines and free fatty acids in SD rats. These results suggest that the intake of caffeine reduced body fat by lipolysis through the release of catecholamines. Catecholamines are hormones produced by the adrenal glands. Dopamine, epinephrine, and norepinephrine are the main catecholamines. This study determined that even at 0.025% caffeine, similar to the level of caffeine consumed in daily tea or coffee use, had anti-obesity effects such as a decrease in both body fat mass and body fat percentage when paired with a high-fat diet. This effect is favorable from the viewpoint of normal daily caffeine ingestion. Levels of epinephrine, norepinephrine, and dopamine also spiked 30 minutes after caffeine administration, before gradually dropping to match those of the control group. There was no difference in the level of serum free fatty acids between the control group and the caffeine administered group at the 30-minute mark, but at 60 minutes and 120 minutes respectively, the caffeine administered group showed significantly higher free fatty acid levels. This indicated that caffeine's anti-obesity effects seem to be mediated by the release of catecholamines, which enhance and prolong lipolysis. Caffeine was also found to inhibit the action of cyclic AMP phosphodiesterase, which increases cellular cyclic AMP and can induce prolongation of sympathetic stimulation and lipolysis. Overall, Hattori and colleagues demonstrated the anti-obesity effects of caffeine, including the reduction of body fat mass and fat percentage in a dose-dependent manner, and concluded that caffeine has potential use as a functional food ingredient that would prevent lifestyle-related diseases in humans who tend to ingest a diet containing a high level of fat. However, this study only specifically looked at rats fed on a high-fat diet. We will demonstrate how this translates to human physiology shortly. However, before we get to that, another interesting effect seen in this study was that following caffeine ingestion, there was upregulation of the uncoupling protein UCP1, which contributes to thermogenesis. Thermogenesis is the process through which heat is produced in humans and animals by uncoupling protons during the electron transport chain of oxidative phosphorylation. It is often mediated by a protein called thermogenin, which uses substances such as free fatty acids to uninhibit thermogenin, which leads to protons leaving the intermembrane space and entering the mitochondrial matrix, bypassing ATC synthase channels and thus dissipating their energy as heat instead. This results in inadequate ATP production in the body, leading to increased metabolism and subsequently lipolysis. In another study conducted by Belza, Tubro, and Astrup, they tested 12 healthy men who were administered with one of 4 tablets: green tea, tyrosine, caffeine, or a placebo containing microcrystalline cellulose. This study observed a 6% increase in thermogenic response after the consumption of a 50 mg tablet of caffeine, but no effect was observed with the other tablets. It was shown in the Hattori study that the subcutaneous administration of caffeine increased the mRNA expression of UCP1 in brown adipose tissues of obese mice. In terms of human physiology, a study done by Atchison and colleagues tested 8 healthy male volunteers. Each subject underwent 4 experiments during which either caffeine at 10 mg per kilogram of body weight or 500 mg of lactose placebo was ingested. In the third experiment, the influence of caffeine ingestion was investigated using the beta-adrenoceptor inhibitor propranolol. Propranolol prevents epinephrine from binding to the receptor, therefore decreasing energy expenditure and preventing the activation of hormone-sensitive lipase, inhibiting lipolysis. This study showed that while caffeine ingestion increased energy expenditure by approximately 13% and lipid turnover and oxidation, beta-adrenoceptor blockage with propranolol decreased caffeine-stimulated energy expenditure and lipid turnover. This indicates the importance of the receptor's role in mediating the metabolic effects of caffeine. Caffeine may also impact the adenosine alpha-2 receptor, which upon coming into contact with adenosine, inhibits lipolysis via inhibition of adenylyl cyclase. Caffeine binds to adenosine, thus not allowing it to activate the alpha-2 receptor, stimulating lipolysis. The large increase in free fatty acid turnover after caffeine ingestion was likely due to the combined effects of caffeine on beta-adrenergic lipolytic stimulation and on inhibition of the anti-lipolytic effects of adenosine. However, although caffeine caused a 2-fold increase in fatty acid turnover, lipid oxidation increased by only 44%, which indicates that most mobilized fatty acids are re-esterified when in resting conditions. Therefore, very few fatty acids are actually broken down. When paired with exercise, however, which does cause an increase in the turnover of fatty acids, 70 to 80% of free fatty acids released are actually oxidized. This indicates that stimulation of lipolysis through caffeine intake alone may not be enough to trigger significant lipid oxidation or energy expenditure, but through combination with exercise may prove more effective than either of those individually in promoting weight loss. To summarize, the direct pathways allowing caffeine to promote weight loss are as follows. Firstly, caffeine promotes the release of catecholamines from the adrenal glands, likely through an interaction with intracellular calcium. This results in an epinephrine cascade, which ultimately leads to the production of free fatty acids through lipolysis. These in turn may be oxidized. Caffeine also acts as an adenosine antagonist, resulting in the inhibition of the alpha-2 adrenergic receptor, which, when active, inhibits lipolysis by inhibiting adenylyl cyclase activity. Lastly, within lipolysis, caffeine was shown to have inhibitory effects on phosphodiesterase, which in turn frees cyclic AMP from inhibition, thus promoting lipolysis. Caffeine intake was also found to induce expression of uncoupling proteins such as UCP1 in humans, which uncouple protons during the ETC, leading to heat production and more importantly, inadequate ATP production. Therefore, the body has to undergo higher levels of metabolism to satisfy the energy demand, possibly leading to weight loss. There are other pathways that we have not discussed in detail that may also play a role, including caffeine's possible synergistic relationship with ephedrine, a weight loss agent. Indirectly,
Caffeine's utility as an ergogenic aid during submaximal exercise, especially its ability to sustain said performance, may be significant in aiding weight loss training. In the future, efforts should be made to conduct more research to discover how caffeine affects the different factors of the mechanism of lipolysis and other weight loss pathways. For example, we can investigate how caffeine increases levels of catecholamine release from the adrenal glands, or attempt to discover how caffeine lowers the oxidation rate of carbohydrates relative to lipids. Answers to these questions can help us with targeting treatments to certain disorders more effectively. For example, this information could be applied to the portion of the population affected by obesity to create more effective treatments like caffeine supplementation to promote weight loss. To conclude, we've determined that caffeine may potentially be used as a weight loss agent to maintain a healthy body weight and help those affected by obesity disorders. Thanks for watching.